here's our gene for GFP in our plasmid, right? There's the GFP gene. Here's the Ara-C operon that switched it on. Here's the ampicillin resistance gene. Here's the origin of replication. Here's the TATA box. That's our P-globe plasmid. So we're basically asking, how did they make this P-globe plasmid? How did they get the gene from a jellyfish out in the Pacific Ocean? Or in general, let's suppose that you wanted to, and we have an assay for the protein. It glows in the dark. You just hold a UV light over it and it glows, so you can detect the protein. Or another example, remember the catalase enzyme, the one that made the bubbles? Put it in with H2O2 and it makes bubbles. See, that's the assay for catalase enzyme. Put it in with its substrate. If you see bubbles, it's evidence that the enzyme is there. And so forth and so on. So this is the case where all you have to work with is you know there's a protein and you have a way to assay for it. You can detect that it's there, and how do you go back to the original organism and get the gene that codes for that enzyme? And again, it should strike you as absolutely amazing that we can do this at all. Well, we can do it in the following way. We've got the protein, and we've got an assay for the protein. Okay? We want to get the DNA over here that's in the original organism, which in this case is in a jellyfish. So it's obvious that if you're going to get your hands somehow on the DNA, you have to allow the DNA in some way or another to actually make the protein, right? Or you never have any chance of getting your hands on the DNA, because you've got to kind of work backwards. So you're going to have to use what we call an expression vector. You're going to have to use some organism to make the protein for you. Say that. We're going to have to use an expression
vector.
In other words, some organism, some living organism that's got ribosomes, it's got amino acids, and it's got all the enzymes necessary to actually make a protein. And you're going to have to somehow get your hands on the DNA and let that organism make the protein. And then kind of catch it in the act, so to speak, if you want to put it that way. That's sort of the big picture here. So you can only do this through an expression vector. And the expression vector that we're going to use is E. coli. So we're going to use bacteria to make the protein from the DNA and kind of catch the DNA, so to speak, in the bacteria by doing some sophisticated things. Now, since we are going to use E. coli here, and ultimately we'll detect it just like we did in the lab. See, here's E. coli, you plate it out on a plate, you put a plasmid into it and detect it with a black light. See, it's glowing in the dark there. That's the bottom line. And since we're using E. coli, we have a problem, which I've told you about. You know that eukaryotes have split genes, right? Say that. Eukaryotes, like a jellyfish, have split genes. And what's true about bacteria? They don't. They have continuous genes. Bacteria don't know what to do with split genes. They don't know how to splice out the introns. You know what happens in a eukaryote. Okay, here's the DNA. Here's these loop domains. See, here's a gene that's shut off. Here's a gene that's shut off. Here's a gene that's on. For what? Catalase. And what happens? The messenger RNA gets made. There it is, 5' 3' N, with the introns and the exons. And then what happens? Enzymes come in and